Our final speaker of the morning is Joe Kilgour, who's an expert inhalation toxicology consultant and the director of Mereside Toxicology Consulting. So before that role, Joe had a long career in the chemical pharmaceutical companies and has experience as a study director and a project toxicologist, and now works with small and large pharma companies worldwide to deliver toxicology programs to support first-in-human studies through to phase 3 and registration. So she worked with drugs in all sorts of different target areas and has been the Association of Inhalation Toxicologists chair since 2011.
Yes, I took over from Paul, so we're measuring my tenure in decades, not years, which I recognise is quite unusual, but nobody seems to want to take the job off me, so I'm still here. So thank you to the organisers and thank you to the delegates for attending this workshop. It's my dubious pleasure to do the anchor session here. So what I'm going to try and do is summarise the talks that my esteemed colleagues have given and give my sort of point of view on what the key messages are, I think. But then also to give you a couple of bonus bits of information as well. Something on novel excipients, which I know some of my colleagues have mentioned, but I want to go into a little bit more depth on that. And also a little bit about intranasal studies as well, because as inhalation toxicologists, as it's part of the respiratory tract, we quite often get dragged into these kinds of projects. And then finally, I'll sort of give you a bit of a brief history of the AIT, who we are and what we do, and then try and sum up with a couple of take-home messages from the workshop. So the first talk from Carsten really sort of set the scene very nicely and showed us that the lung has been a target for therapy for centuries. I'm not sure we were quite getting it right until probably the latter half of the 20th century. Cigarettes, etc., not really being what we're aiming for now. Although I think we're on a track for repeating history with vaping at the moment, but we'll see what happens there. He talks also about the advantages of this route of exposure, bypassing first-pass metabolism and reducing systemic side effects, but also the disadvantages as well, how technically difficult it is and expensive. Complex projects, you've not only got your formulation and your drug, but you need to choose a device in order to deliver it. There are patient compliance issues, and also, once you've gone through all of that, you might not even get reimbursed by the health authorities if there isn't a significant advantage of your drug over existing therapies. But despite these difficulties, people are still wanting to develop inhaled drugs, and that keeps people like us in a job. So I can't complain. I guess one of the things he did point out as well was that whilst we spent most of the 1970s up to the 2000s focusing on respiratory diseases and a very few number of them with not very much innovation, we're now looking at more complex diseases and different targets. And IPF is certainly one of the ones that comes to mind. And unfortunately, Paul didn't get a chance to talk about the bleomycin model, which is one of the ones that's used for projects in that space. So then we moved on to in vitro, which is something that's quite close to my heart. I started my life as an inhalation toxicologist as a PhD student, developing a nasal respiratory epithelial model with actually one of the founders of the AIT, which I'll come on to talk about later, as my PhD supervisor. And I think what Vicky told us was that there's a whole range of models from the simple to the complex, and that reflects really the complexity of the anatomy and physiology of the respiratory tract, I think. And then you have a choice between submerged culture, air-liquid interface culture, and variable lifespans of all of those models. And she really highlighted the importance of choosing the right model for the job. So there isn't a one-size-fits-all, and certainly, you need to be very careful in choosing the model that you use to look at the question that you're trying to answer. There are multiple endpoints that can be studied there, and there were many and varied, but one of the ones that really translates back to in vivo for me is the histology. So any of the models that enable you to have that anchor between in vivo and in vitro studies is important for those correlation studies. And then there's the dosing and dose translation aspects that she talked about as well. So moving on to Tony's excellent talk about the actual hands-on process of generating the atmospheres and exposing the animals to them. The full range of different physical forms of test material. Gases is an unusual one, and we don't do that very often in pharmaceuticals, but of course, recently there's been the push to reduce the global warming potential of propellants, and certainly that's been a hot topic recently that I've been involved in. Basically, all we can do as inhalation toxicologists is generate an atmosphere and get the animals to inhale it. But in order to do that and know that we're getting the material in to where it needs to go, and to be able to calculate those doses, we need to really characterize those atmospheres very carefully. And I think that came across in both Tony's talk and also Paul's as well. So atmosphere characterization is key to enable us to use the algorithm that we use for calculating the doses. We can expose pretty much all of the key tox species, and we went into some detail about that, but one of the key take-home messages from Tony's presentation, and I guess also Paul's, is that training and habituation of the animals is of paramount importance. It is quite a stressful procedure for the animals, and if you don't do that training to keep them calm during the studies, then the data you get from the studies is not going to be that great. And then Paul's talk went into much more detail about the study designs and so forth. They're largely guideline-driven, both for pharmaceuticals, which we focused on, but also, I guess, for agrochemicals and chemicals, which is a sector I've also worked in. But there are sort of differences in how we do those studies, and I think Paul highlighted that too, in terms of we're trying to minimize the duration of exposure for pharmaceuticals, whereas you're having to go to guideline-derived, much longer exposures for chemicals and agrochemicals. I think both Paul and Tony mentioned that these studies tend to be very expensive, and they're demanding high levels of high amounts of test material. And it's good to incorporate those safety pharmacology endpoints on these inhaled tox studies if you possibly can, because they are so difficult to do, because they're expensive. And it does give you that advantage of being able to monitor for effects of repeated dosing, which you wouldn't pick up in an ICH S7 guideline study, which is just a single dose study, and actually doesn't actually have to be by the intended route of exposure. We use these studies then to derive safety margins. We have, regulatory authorities expect us to demonstrate the safety margins listed here. So 10-fold and 5 to 6-fold safety margins to local exposure in the lung for rodents and non-rodents respectively. But also we have to look at the systemic exposure as well, and that needs to be about 1 to 2-fold for systemic safety as well. And interestingly, there's no consistent approach between regulatory authorities on how to actually do this. So this is dictated really by FDA, who are the most conservative of the regulatory authorities, and we then use that information to generate or recommend a maximum starting dose for those first-in-human studies that Paul was talking about. And actually, I'll come back to this point about the lack of consistency when I talk about the work streams that the AIT are running at the moment. Okay, so those are the take-home messages as I see them from the very good talks that have gone before. So I'm just going to add a couple of points now, which I hope you'll find of interest. So in terms of novel excipients for inhalation, Paul mentioned this in his talk. Quite often, we use fairly standard vehicles, so those could be buffers, citrate and phosphate buffers, saline for liquid formulations, or lactose or magnesium stearate as an excipient for powders. And of course, there's the FDA inactive ingredients database, which lists all the approved excipients for use by inhalation and other routes of exposure as well. So you can go to that and have a look if you're designing a new formulation and make sure you're within that. But there are, and I probably don't need to tell this audience, but there are sometimes situations where you need to add a novel excipient for various reasons. So that might be, let's say, for example, for stability reasons, you decide that you're going to produce a lyophilized powder for reconstitution for your drug. You might need to add something like mannitol to stabilize the lyophilized powder. You might want to be getting better solubilization of your API by using something like a cyclodextrin. So you can use an aqueous formulation and also then reduce the amount that you have to give and therefore hopefully the side effects. You may need to add surfactants to improve the performance of your material in your chosen clinical device. Or you might just want to enhance permeability or reduce clearance by adding something like hyaluronic acid to a formulation. And when we talk about novel excipients, that can mean not just a different type of excipient that isn't already approved, but also a higher level than is in an approved formulation on the market already. And there are regulatory guidelines for excipient qualification, which you would need to do if that was the situation you were in, that you can look at, and I've got that on the next slide. But it's important to note that if you're wanting to market your drug in the US, even if there's a novel excipient an excipient that you want to use that's been in a marketed drug in the EU for decades with, hundreds of patients, thousands of patients have used it for decades, that doesn't matter. You still have to qualify it for use in the US. And I've been in a situation like that with one of my projects. So what do you need to do if you need to qualify a novel excipient? Well, depends on your duration of use for your particular drug, but if it's for long-term use, then generally you'd need to do a 6-month rat toxicity study, and that needs to include that air control that Paul mentioned. I had a situation where I joined a project partway through. The 6-month tox study was already running without an air control, and we had to do a further qualification study on top of that because we didn't have the air control in the 6-month study. We were, however, able to negotiate with FDA and just do a 3-month qualification study on the basis that we had the 6-month study already. It's also possible to look at qualify multiple novel excipients in a sort of to-be marketed formulation. You don't have to do them all separately. And I've got the guidelines sort of on the slide here for you. But I would recommend for developers to keep it simple if you possibly can. It makes everybody's life a lot easier. And if in doubt, do interact with your relevant regulatory authority and get some advice before you go into studies. So switching tack a little bit now to intranasal studies. As I say, we sort of get dragged into this as inhalation toxicologists quite often, and there are some peculiar considerations that you need to think about when approaching these studies. So species differences in anatomy of the nasal cavity and the size of the aperture will determine what dosing method you actually use, and that might be just a pipette or a syringe or a microspray, and we'll see some pictures of those in a moment. Also, the maximum volume that you can put in per nostril, per occasion, and per number of occasions per day will also limit the total dose you can give in a day. The nature of the test material might be a bit tricky to handle as well, so again, you'll probably need to do some experimentation to figure out what the best dosing method is. And quite often, nasal clearance can be quite fast. We heard about all of the mechanisms of clearance in the previous talks. And so this presents a challenge in terms of being able to detect the systemic exposure in these studies. And as with inhalation studies, the local histopathology in the nose is key, and also in the brain because of the potential for nose-to-brain transport that can occur. And FDA, for example, will be looking for a precise number of levels of the brain to be examined in these studies. The metrics as well for working out safety margins here are different to those for inhalation studies. So rather than, we heard about the margins just before, well, we would work those out on a mg per gram of lung basis normally. Here we're going to be working with mgs per centimetre squared of nasal surface area. So that's how we do it for this. And the safety factors that we need to achieve with these studies is lower, so it's 2 to 3-fold. And that's probably a reflection of all of the restrictions that there are in these studies in terms of trying to get the dose in. So here's just a quick picture. I hope you can see that on the screen. It's not too bad. Just showing a sagittal section of human versus various species in terms of the nasal cavity complexity. And then here you've got the transverse sections of the rat, the 4 sections that we would normally do for an inhalation study. And then on the right is a table with the volume, the surface area, and the turbinate complexity listed there. And the 2 references for these are at the bottom of the slide here. But it's important to note that in terms of the structure of the nasal cavity, anatomically, the human and the monkey are the most similar. For the other species, the complexity is a reflection of how important the sense of smell is for the survival and function of the animal, if you like. But in terms of the total nasal surface area, actually the dog is much closer to human. And here, just quickly, a few pictures. The one just here is probably familiar to most of you since COVID, the old flu and cold nasal spray that you might have from Boots or whatever. And, you know, we have syringes, micro sprayers, and some of these devices can be modified slightly to enable them to be used for rodents. We tend to use pipettes generally, or the modified Aptar kind of device there for rodents, and micro sprayers generally more for the larger animals. And then I'm not going to go through this, but there's a whole list of factors here affecting nasal absorption in experimental animals. And I just wanted to point out where the arrow is, loss anteriorly from the nose. So this is relating to what I was saying earlier about the limitation on the volume of material that you can get into the nasal cavity. If you put too much in, it's going to either flow back out of the front or get down the nasopharynx and swallowed. And so that's a pretty pointless exercise. So you have to work within those limits. This is just a, installation versus insufflation in terms of the different methods. So we'll skip through that pretty quickly. So just to summarize those 2 points that I've covered then, excipients for inhalation, what I would advise you to do is keep it simple if you possibly can. From our point of view, that makes life a lot simpler. If you're going to have to add additional components, make sure whether you need to qualify those with specific type of study. And if you're at all unsure, take advice from your relevant regulatory authority. And for intranasal studies, you have these quite significant limitations on the doses that you can deliver. And so you have to account for that versus the doses that you want to give in the clinic to make sure that you can achieve those safety margins, as with inhaled studies. Okay, so take a breath. In the style of Professor Stephen Hawking, I thought I'd give you a brief history of the AIT now to explain why we're all here and why Ben invited us to run this session with you today. It's not going to take 3.7 billion years to go through this, and it's hopefully going to be slightly less complicated than that book. So the AIT really was created by 3 founding members, one of whom, as I said, was my PhD supervisor. And they had their first inaugural meeting in 1981. The key criteria that they wanted was that members should be actively practicing inhalation toxicology, and that then we could have a meeting each year to discuss, key aspects of what we're doing and try and keep each other all appraised of advances and so on. And those meetings have happened once a year, every year, apart from, obviously, during the fallow years of COVID. And as a result of all of the skill sets that you've heard about today from all of the speakers, the members really have many different skill sets due to all the scientific disciplines that are involved in running these studies. So, as I say, we've had a conference every year. We tend to now, unlike DDL, which very sensibly holds its conference in the same place every year, which makes life really easy, we like to make our life difficult by rotating on a 3-year cycle between the UK, Europe, and the US. So this year, post-COVID, for the first time, we met in Gothenburg in Sweden. And next year, we're going to the US, and I'll tell you a bit more about that in a moment. Topics vary from year to year, but we like to keep it, current hot topics mostly, and then there are other ones that we revisit on a regular basis because you make advances as time goes on. So in vitro, obviously, is one that's come around. Nanoparticles, e-cigarettes more recently comes up on a regular basis. So that's what I want to say there. And so in terms of our membership, so we don't charge a membership fee. If you come to a conference in person, you automatically become a member for 3 years. And so because of that fallow period over COVID, our number here of current members just reflects the number of people who attended Sweden this year. But we have had up to a rolling number in about the 300s, which is not bad for a highly specialised group of individuals. So we hope to get those numbers up again soon. And so what do we do? What have we done in the past and the future? Obviously, I've already mentioned we have an annual conference. And we have tried to forge links with other learned societies during that time. So notably, we had a proper joint meeting with BSTP, the British Society for Tox Path, in 2016. And as you'll see, some publications were a consequence of that collaboration, and also with ISAM as well in the US in 2014. We have produced some publications, and Paul's already mentioned one that you'll see quoted on pretty much every inhalation tox study that comes out of a CRO to help us calculate the inhaled doses. And I'll show you a few more on the next slide as well, but we're currently in discussion with BTS, British Tox Society, about a possible peer-reviewed paper in their journal, ToxRes, and also maybe a communication that just goes on their website. So those discussions are ongoing. And I just want to talk to you a little bit about some of the work streams that we're running as well. So there are some grayed out ones here, which I won't go into too much detail about, but Paul mentioned time versus concentration. We can vary those to achieve a dose, and we believe there are certain situations which you would vary the concentration and not the time, but there are situations when you do it the other way around. And the PSD one relates to something Tony mentioned about, calibrating those cascade impactors and how we do that. But again, I'm not going to go into that, excuse me, in much detail. So here I'm just highlighting the Alexander et al. 2008. We've already talked about, excuse me. Got a bit of a cold going on here. The 2021 paper at the bottom was actually coming out of that collaboration with BSTP in 2016. This is a technical paper about the actual conduct of biological inhaled studies. And there's a sister paper to that which focuses on the pathological aspect. And this was an industry survey that we ran, which looked at about, I think, 12 different studies that companies were willing to share with us on an anonymized basis. And we did some analysis of that. And those two papers were published in a special edition of ToxPath. So we tried to be quite active. I've also put Ben's paper in there, which, although it wasn't an AIT paper per se, there were quite a few of the AIT committee members and members involved in that one as well. So, excuse me. In terms of the work streams that I did want to talk about in a bit more detail then, the regulatory harmonisation work stream. So this goes back to what I was saying about the lack of harmonisation in terms of how to calculate those safety margins for inhaled studies. Currently, there's no specific guidance, but everybody defers to what FDA would like us to do, which, as Paul was saying, is slightly unrealistic in that it assumes 100% deposition in humans, which we all know isn't true. And so it creates a problem if companies start to develop their drugs outside of the US and then want to go in and can't achieve the required margin. So our aim with this, and there are lots of people talking about this in other fora, such as ACT, American College of Toxicology, and so forth, where we want to work together here and put some sort of communication out, hopefully a peer-reviewed paper, position paper, just to get the conversation going with the aim of trying to move some way towards harmonising the way we do things. Excuse me. goblet cell hyperplasia in action. Okay, and the next one that I just wanted to highlight is the NAMs for inhalation workstream. So we're quite excited about this one. Very current, and obviously Vicky's interested in this one. But we have Annie Yarabek, who recently joined the AIT committee. She is like a train. She's got so much enthusiasm, and she's really driving this forward, and has been pleased to be part of the AIT because she can see how the skill sets that we have can really help with what she sees as an area around NAMS that perhaps isn't being given the attention that it needs. And with that, I'm talking about the dosimetry, the characterization that we've all talked about as being of critical importance for inhalation studies. Well, it's also of critical importance for any of the data that's being built into some of these computational models. And so that's what we're aiming to do with this work stream, is to forge links with groups like the ASCCT and try and have some influence there with this. So that's the ambition for that one. Okay, I think we're nearly there. Excuse me. So Carsten got his plug-in at the end of his talk, so I'm going to do the same for the AIT here. So we're currently planning our next conference in September next year, so it will be the week of the 16th. We're currently just negotiating with the hotel on the final details of this, but it's going to be in Cleveland, Ohio, hosted by Charles River out there. They're going to help us run this whole show, and the suggested title is Inhalation Toxicology Rocks the Respiratory Sciences, or maybe just Inhalation Toxicology Rocks. And I'm certainly hoping to get to the Rock and Roll Hall of Fame to see some of my heroes in there. More importantly, though, the topics that we're going to discuss at that conference are listed here for you. We've got 2 sessions on NAMs, the ones on the left-hand side there. We'll return to inhaled biologics again with a bit of an update on sort of gene therapy and cell therapy, hopefully in that one. Expand on intranasal studies, the sort of discussion I've had here with you, go into a bit more detail on that. Aerosol generation has always been a drive and a feature of AIT conferences, is to keep on top of any new developments there. But also then, because traditionally, and certainly when I first got involved with this group in the sort of early 90s, I guess, I was working in agrochemicals and chemicals at that time, and I found that there was very much a slant towards pharma with these conferences. We're trying to even that out a little bit, and so bringing Annie on board from EPA has really helped with that. And actually this wildfires and smoke inhalation toxicology session was a suggestion of hers, and I think it's very topical and also quite interesting and a bit different for us to understand what's going on in that area as well. So that's what we're going to be doing next year, and I'd be really happy to see as many of you there joining us for that. And so just finally then, take home messages as I see them from the whole session today. The importance of aerosol characterization in the studies that we run, in vitro, in vivo, whichever, because only then can we estimate the dose that we've delivered accurately. So characterization, characterization, characterization. Inhaled preclinical studies are technically challenging. You'll know this because there aren't many CROs in the world that can do them to what I would consider the required standard anyway. They are expensive because of all the pre-work that's done, because of the experience that the technicians need to have to run them well. And of course, they require a lot of test material as well. That can be a shock to clients, particularly if we're talking about biologics as well, because those test materials tend to be quite costly. And then, last word, keep inhaled formulations as simple as possible, and that does make our life a lot easier, and yours. And with that, I'll say thank you very much for joining us.